Lots of students find this a tricky question because the processes are so confusing. The correct answer is, of course, that RNA polymerase doesn't care about stop codons at all. Stop codons are signals that are read in RNA by the ribosome. RNA polymerase sees stop codons as just like any other component of the DNA, and it just keeps going. Now, what we've done, we've talked about genes, that genes are basically informational entities in our DNA. Sometimes they specify functional RNAs, like parts of the ribosome. Most of them specify messenger RNAs that code for proteins. And genes are identified by the cell by the presence of regulatory signals, short sequences in the DNA that tell RNA polymerase to make an RNA copy, and short sequences that act in the RNA to tell the ribosome where to start and stop to make a protein. Coming up next, we're going to talk about how this coding for proteins actually works. I hope to see you there.